why cancers develop really didn't have a component of what the microenvironment does, but it turns out that every aspect of what Hanahan and Weinberg say are important for cancer development is supported by the microenvironment. So yeah, I won't go through each of the different things here, but every single thing in this circle requires a support of the microenvironment to grow. So I'm going to tell you a little story about a protein that we're interested in, and I'll take you through first how we got to this point, because quite honestly, it was a very serendipitous sort of a finding that this protein works on the microenvironment. Originally, actually, this protein was, we investigated this protein. I'm not getting in advance. We investigated this protein because there was a lot of amplification in head and neck cancers at 3Q. We did a positional cloning analysis to narrow down that area. Please advance. And ultimately, we cloned out this gene, and this was the human genome days. So actually, this didn't exist as a gene in the database. We had to do annotations and a few different measures to prove first it was a gene, and then after we proved it was a gene, we had to then prove that it was an oncogene, and we did that both in vitro and in vivo using several different models. Even transgenic animals, this was able to produce cancer. We went on to determine its cellular activity by using knockout mice and Drosophila models to see what it was doing at the cellular level, and we were able to really tease out how it functions at the biochemical level. But despite all of this, there was still a lot of question as to whether this is something that is a target of the amplification that was present in 3Q. And a lot of my grants, when they came back rejected, said, well, there's no proof that this is actually a cancer driver. And there was a lot of reasons to think that it is a cancer driver. So if you look at the TCGA data set, you find a lot of amplification in SCRO, and this is across just about all of the different TCGA projects that are out there. The red lines, the dark red bars are amplifications. If you see there are rare mutations, they are actually activating mutations. They're relatively uncommon, but the mutated forms of this protein are much more oncogenic than just simple amplification. So clearly this is a cancer driver, and what really made us happy, there's the usual correlations between copy number expression. There was a strong correlation on the TCGA data set for head and neck for survival. What we found interesting is, for example, breast cancer had only 4% of cases amplified, but the survival difference was massive. There was a 55-month survival difference at 3 years, which was the median that was available in this data set. So whenever this gene was activated, there was a very aggressive phenotype. And so what made us, sort of get into the mainstream, if you will, or start to get into mainstream a little bit, is a couple of papers. The first one was this analysis of the lung TCGA, which analyzed the 3Q amplicon, and through a series of bioinformatic and then experimental assays, they came out and said that the best target at 3Q is indeed this SCRO protein. And then the second big thing for us, next, is a cross-TCGA analysis that tried to find the most druggable genetic abnormalities in all of the TCGA data sets, and this came out as one of the top 10. So this validation kind of spurred us on to try and figure out how we can potentially target this gene and really establish whether it really is a player in cancer. And the way we, next one please. The way we did that is we made a knockout mouse and then asked this very simple question: is this gene required for cancer formation? And the easiest way to make oral cancers is to give 4-NQO to these animals. We fed them. Sylvia Goodkind helped us with these experiments. And lo and behold, as we fed them, next one please, we found that the knockout mice Next one, please. Had both fewer tumors and smaller tumors. So these tumors actually didn't grow very well. They never actually caused the death of the animal, whereas in the wild-type mice, the death was invariable in all of these mice. So there was clearly some effect happening at the level of either the cancer cell or the microenvironment. We weren't sure. We thought it was a cancer cell. But when we looked under the microscope, these tumors looked more different in the microenvironment or the surrounding environment around the tumor, with a lot less desmoplasia. There was more lymphocytes infiltrating. It looked dramatically different. And what that led us to do, next slide please, is to ask the question if indeed it's a tumor cell or the microenvironment. And what we did is we took syngeneic tumors, which were wild type first grow. Some of them were amplified, some of them were not. And we put them either into knockout mice or into wild type mice into the flank. We chose melanoma just because we could see it and it's a lot easier to find them in different locations. And it turned out that, I don't think I have a pointer here, do I? If you look, the bottom one is the knockout mice. You see the black. It's basically flank injection on either side. This tumor has grown through and through, and that was the typical characteristic. No, this doesn't have it. Oh, okay, sorry, I got it. This was the typical characteristic of these tumors. They tended to have very little necrosis, and they were rapidly progressive. They even developed metastases. Whereas in the scroll knockout tumors, we barely had any tumors get beyond the flank wall, the skin, or the subcutis. Only one tumor actually infiltrated into the musculature. None of them did this sort of through and through growth. And when we started to look at these tumors under the microscope, there was a massive amount of necrosis that was developing in the knockout animal compared to the wild type animal. This happened not only in melanoma. We did lung, we did breast, we've done mesothelioma, we've done ovary, and really it didn't matter what tumor it was more about the microenvironment than the tumor type in this case. And as we start to dissect these a little further, what we found is a clear difference in size. There was difference in the local invasion, and also some suggestions that there may be differences in metastatic sites, lung being the primary metastatic site for these melanomas. And the amount of necrosis, so the blue dots here are the knockouts, and the red dots are the wild type. The tumor size is here and necrosis here. Most of the knockouts tended to be to the smaller tumor size, but yet tended to have more necrosis. Even with larger tumor size, except for this one outlier, these wild types had much less necrosis on average compared to these tumors. When we looked at microvessel density with MECA32 staining, there was much less angiogenesis in the knockout compared to the wild type. When we looked at infiltrating lymphocytes, the knockout versus a wild type, I mean, every single field looks like this. Doesn't matter what the size of the tumor is. It matters more which animal it was in. And just to kind of confirm that, this is what it looks like when we did flow on the knockout versus wild type tumors. The CD45 cells are quite different in these tumor types. We wanted to see if this actually did affect distant metastasis, so we did a tail vein injection in the melanomas, and this is what the wild type animal looks like every single time, and this is what the knockout animal looks like. Rarely, if ever, was there any uptake of tumor at any site, no matter where we were looking. Lung and liver usually are the most common, and you can get subcutaneous metastasis as well, but you never got that in the knockout animal. So clearly, the microenvironment was important, and it was affecting many different things. And there's multiple different possibilities of what could cause this, but this central effect made us think that it may be a central player, and some of the most important players in the microenvironment come from the bone marrow. So what we did then is we repeated the experiment. You can see the knockout animal with the small tumors in the flank, the wild-type animal with the tumor infiltrating through, and we did a bone marrow transplant in the knockout mouse and put in wild-type bone marrow. And basically, this rescued the growth of these tumors. We got much larger tumors, much less necrosis. The amount of necrosis was about the same as a wild type, whereas the knockout had much more necrosis, and the tumor growth rate was clearly rescued in these animals. So by not changing anything but the bone marrow cells in these mice, we were able to rescue the growth of these tumors, and the metastatic phenotype and everything else also followed suit. Well, there's two main components within the bone marrow. There's a hematopoietic stem cell component, and there's a mesenchymal stem cell component. So we decided to tease that out a little further. First off, by the way, if we take wild type mice and we transplant them with knockout bone marrow, they have the same phenotype as a knockout animal. So clearly it was the contribution of SCRO to cells within the bone marrow that was giving this phenotype, and we can do this repeatedly to know that this is what's happening. So whole bone marrow transplant rescued growth. It's really hard, it turns out, to isolate mesenchymal stem cells from the bone marrow. So what we did instead is we isolated the hematopoietic stem cell component and did a transplant of just the hematopoietic stem cells, and actually that could not rescue the growth. And the growth curve is just shown here, basically just expanding this into larger numbers, and the necrosis correlated with the size of the tumors and the predicted outcome that we were thinking. Well, we did have access to our wild-type mesenchymal stem cells, and it turned out that if we gave the knockout animal wild-type mesenchymal stem cells, it actually now grew at the same rate, and the amount of necrosis dropped dramatically. So we are quite confident that it's the mesenchymal stem cell component in the bone marrow that's driving this environmental contribution to the SCRO knockout animal. So at the end of the day, there's some important keys here. These are very stable cells. They're not genetically altered. They're functionally altered, but they're not genetically altered. But these cells play a role in many different activities. So mesenchymal stem cells are essential for wound healing, for example. So if you disable a mesenchymal stem cell, your wound healing, your ischemia reperfusion will also be very dramatically abnormal. We didn't see any gross differences in these mice, but we went ahead and I'm going to go ahead and skip this. These mesenchymal stem cells do a lot of things, and hence the reason for the effect on healing. We looked at ischemia reperfusion injury, and we looked at a skin wounding assay, and there was no difference. So what was happening at the tumor level was not happening to other activities of the mesenchymal stem cell. It was restricted to the host tumor interaction, not other things that these mesenchymal stem cells were doing. So what this leads us to suggest, and it's far from conclude, but definitely suggest, is that there are activities of these mesenchymal stem cells and probably other components within the microenvironment that are unique to the tumor environment. And their other activities are not compromised, even when you remove what looks to be an essential gene for the tumor microenvironment. So this really buoyed us quite a bit, and we're in the process of, we have compounds now. We've completed a high throughput screen. We've organically synthesized into the 4th round of new compound, and they're working at the nanomolar level, so we're looking to recapitulate the genetic phenotype that we saw here with the drug effect. But what this tells us is that maybe we can actually dual target these cells. I mean, maybe SCRO is unique, maybe it's not, maybe there's other things that do this type of thing. But certainly, if we target SCRO, we're going to get effects in cancer cells. There's an oncogene addiction phenotype. So when SCRO is upregulated or overexpressed, and you knock down the protein, cancer cells die. But we may actually get a prevention phenotype or a microenvironment phenotype when the tumor is already there. So we're very excited to bring some drugs forward. Clearly, if you think that we have some understanding of the cancer genome, which we probably have very little understanding of the overall picture, we have even much less understanding of the tumor microenvironment. So to move forward, we really need to understand what's happening at this microenvironment method, at the microenvironment level. And why is that? Well, these are not hypermutable cells. These are genetically stable cells, and they're essential for the tumor. And the one thing that I feel pretty comfortable in saying is that when you change the microenvironment, there's many other models of this, but the scroll knockout mouse tells you that you change the tumor growth. You change the tumor metastasis, you change the tumor behavior, and you even change uptake of the tumor at distant metastatic sites. So you have a dramatic effect that won't generate resistance because these cells are genetically stable. So as opposed to cancer cells, where you have this hypermutability and this chances for resistance developing, the microenvironment won't. And this PD-1 data that I stole from Jose Baselga probably is a great example of that. So clearly the target is a component of the microenvironment. It's the immune system, its contribution to the microenvironment, and these sort of response plots in head and neck cancer start to suggest that targeting the microenvironment shows you pretty good results. And so as we move forward and think about the title of this panel, I think we need to think past just thinking about the cancer cells and start thinking about the microenvironment, and probably the next level is the interaction between the two. So I thank you very much for your attention.